Hi everyone, today I am here to discuss about genetic and breeding tools used in poultry industry. First, if we analyze the title of this presentation, what can we see the key aspects? There are several key aspects here. One is genetics. Genetics mean genome, genes, inheritance, selection markers, or genetic engineering concepts. What about the breeding? Breeding means it's some sort of crossing, maybe inbreeding, outbreeding, species hybridization, breed hybridization, etc. And some aspects is reproductive biotechnology, using the artificial insemination, in vitro fertilization, embryo transfer, germ cell transfer, or stem cell technologies. Another aspect is application in commercial poultry farming. So what are the applications of these breeding and genetic tools? Normally to increase the productivity and efficiency in the farm, or maybe to prevent and reducing the mortality, or maybe improving the quality of the products, quality of meat and eggs. Here, I'm showing you some uses of the genetic tools in poultry industry. I'd like to give you one example. In the poultry hatchery means the segment of poultry industry that produce the baby chicks. And these baby chicks are bought by the broiler producer or laying egg producer farmers. But when the layer farmers buy the day-old chicks, they only want the female chick. They don't want the male one because the egg comes from the female. And basically, no need the presence of male with the female to produce the eggs. That's why the hatchery businessmen need some technology to separate the female chicks only at the earliest stage, at the day old. Let us see what are the techniques used to separate these male and female chicks at day old. Commonly, we use 3 sex-linked genes, namely silver and gold coloring genes, barring and non-barring condition genes, slow and fast feathering genes. I'd like to give you one example in details. When you go for silver female and gold male crossing, the genotypes for the sex chromosome is, for the male, sometimes we call the sire, is the homozygous, Z and Z. For the female, we call the dam also, is the heterozygous, is Z and W. So when you do the crossing, actually what happens? In the male, as the genotype is the homozygous, both the chromosomes are equal of size. But in case of female, it is heterozygous. But not only the heterozygous, there is a difference in the length of the chromosome. W chromosome is much shorter than the Z chromosomes. So that's why in the extra part of the Z chromosomes, some genes might not be found in W chromosomes. Actually, the trick is here. The silver and gold color genes present on this part. That's why when this male silver having the capital S dominant for producing the silver color, as we can see in the slide, cross with the gold that is carry the genotypes small s, they produce 50% female and 50% male. Half, silver color and red color. When we see the genotypes, actually how can we identify which one will be the silver color and which one will be the gold color? As you can see here, The right-hand side one is the female. And the technique to identify is it has darker dorsal stripes on their backs and up onto their heads. But in case of male, then that is left side one, have lighter stripes and it never reaches to head. So if we summarize this breeding and genetic technologies used in poultry industry, we can say in poultry, the use of breeding and genetic tools are mostly visible, common, and it helps for the quick improvement in poultry genetic resources. So that's all. But before ending my presentation, I'd like to acknowledge the source of photographs, some open access online resources. So that's all about my presentation. Thank you so much. See you later.